M5. M5 is all about how is the structure of a protein related to its function, and firstly, how does it get that structure, and then how could that possibly be related to its function. That comes up a lot. M5, structure of proteins. And function. How do we get a protein jar?
Using the ribosome to make a polypeptide chain.
How do we know the order of the polypeptide chain?
From the mRNA, which comes from the DNA.
Excellent. So let's do that. So I'm going to draw the nucleus. I'm going to draw a segment of DNA, which is a gene, a very small gene in this case. It's been opened up. Okay. We've got a template section on the top that we're going to make our complementary mRNA sequence from. So in this case, the particular sequence is A, T, C, G, A, T, C, C, A. So we're going to make a complementary mRNA from that. Zane, what would be the complementary mRNA for that sequence?
mRNA. U, A, G, C, U, A, G, G, U.
Excellent. There's our mRNA. Complementary in antisense, meaning it's running the other way. We've created it using 5' to 3', so we're having our nucleotides come in and make a complementary sequence to that template strand. Okay? What's one of the reasons why we make an mRNA strand, Jamie? One? Excellent. Reese, what's another? Why not just use DNA? We don't need uracil. We've already got the message. We can, if we can read DNA, we can read DNA.
Too big to get out.
Too big to get out. Too big. This is only a small section of a massive DNA molecule. We only want that small section, so it's going to go out. Okay? That process from here we know is transcription. Okay? Now we go outside into the cytoplasm and we're going to form the translation component. As the ribosome moves along, we have our transfer molecules bringing in the correct amino acid. How do we know we have a correct amino acid, Luke?
Because it's got a 3 codon.
It's got the 3 nucleotides on the bottom, which we call a what? Anticodon, which will pair with the 3 sequence codon. Okay? When 2 adjacent amino acids get close to each other in the ribosome, we form a peptide bond.
Is that the same as the hydrogen one? We can hold those up.
No.
So where does the hydrogen bond occur?
A hydrogen bond occurs basically between different parts on the peptide molecule as it gets bigger. So a hydrogen bond, correct me chemists if I'm wrong, there's an attraction between hydrogen and oxygen atoms. Is that right? And nitrogen fluoride. This peptide bond is actually a bond that forms a water molecule. Okay, so actually releases a water molecule. It's a much stronger bond than a hydrogen gas. Okay? But we can go into a bit more detail on that as we go on if we like. So we then need to organize that structure of an amino acid, of the polypeptide chain. Okay? We have the first order we call So this is the order of proteins, the structural order, the hierarchy of protein, the organisation of protein, all of those words meaning the same thing. So you could say the order, the organisation, all mean the same thing. So Jai, put your MacBook down. Okay, it's all in our brains now. We've got our trigger words. We should be able to remember everything. Okay, what's our first organisational level of a protein? Primary. Yeah. So we have our primary organisation of a protein. What does that entail?
Just the polypeptide chain.
Just the polypeptide chain? More than that.
The order, the sequence.
The sequence, thank you. So this is the
One word, Andrew. No.
sequence of amino acids forming a polypeptide chain, or polypeptide. Okay? That's the primary organisation. Just the sequence of amino acids. That's it. How many different sequences of amino acids could we have? Infinite. Infinite. What determines the sequence of amino acids? mRNA and then DNA. So the sequence is DNA, mRNA, polypeptide. So if we want a different polypeptide, what do we have to do? Change DNA, read a different section of DNA. So as we get more and more involved in genetic engineering, if this is the language of life, all we have to do is change that DNA sequence, we can create different proteins. Once we have the ability to know exactly how every single gene is expressed and regulated and what it makes and how that functions together with other things, we could technically, if we have the ability to code our own DNA, we can make any species we want. We can make brand new species. Because I just get on my computer, like I'm playing Sims, and go, I want blah, 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 blah. I want wings, I want legs, I want big arms, I want a big T-Rex head. And I just code it in and inject it into an embryo, let it grow, and it will express my DNA, make the proteins that I want. Next minute I have a T-Rex flying zebra. Because I can. Is it ethical? That's up to you guys to decide, because you're the scientists that will be doing that type of thing. Okay? Eventually a polypeptide will become a protein, but DNA gene doesn't necessarily have to make a protein. It could make mRNA, tRNA, rRNA, microRNA.
Just twist the camera so the title instead.
I've got the fisheye lens, I reckon.
Oh, really?
You can see everything. I don't know how much detail. Let's give him a little flyby. Meow.
That's so good.
Hope my skin looks good on this camera. I do moisturise. Okay. Next organisation level. So we've got our sequence. Matt at the back. Considering you haven't done it, you need to be listening really carefully, and you should know what's coming up just by reading my mind. What have we got next? Primary. What comes after primary? You're in it now. Secondary. I probably should be using red to start as well. Secondary. Okay, the secondary structure has two main structural components that help it start moving into a three-dimensional shape. Does anyone know one of them?
Is it the weak bonds?
It is weak bonds.
Yeah, because curves.
Coiling and
Coiling, folding, that's tertiary. We're not quite there yet.
Oh, is it attractions and repulsions?
That's happening in tertiary as well.
So we know tertiary.
So, before you start thinking that this is one process, so it goes primary, secondary, tertiary, quaternary, it's happening instantaneously, but there are distinct, very minute stages. So in secondary, we have two main structures forming. We have alpha helices. Sorry, that's not a proper alpha. Alpha helix, and they look like these. So these are nucleotides forming a coil. Okay? And we have theta, beta, pleated sheets. And they look like this. Okay? So if we were to draw that secondary structure, we're going to have something like this. Okay? And these are all just amino acids. So on. Okay? So the amino acid chain forms these alpha helices and beta pleated sheets. Beta pleated sheets. Beta pleated sheets, something like that. Okay? Alpha helices and beta pleated sheets. Did my PowerPoint tell you what they're caused by?
This is the structure for each amino acid.
There's basically there's some bonding going on here. Okay. Now, if we remember our amino acid, we've got 20 different ones. The only reason why they vary is because of their R group. So basically, 3/4 of the protein, 3/4 of the amino acid is exactly the same. But we have one section of the protein that's different in all 20. Because of that different R group, you get all these chemical interactions. Okay? You get some bonding happening that causes them then to finally get a three-dimensional shape. Okay? So after primary, secondary, we then go into tertiary, just like your schooling. So tertiary education is university. And in tertiary, this is where we form the final three-dimensional shape. And it's caused by spontaneous folding, coiling, attractions, and repulsions. So you might have additional hydrogen bonding, you might have, what are they called? Van der Waal interactions, I think they're called. I have to look that one up for spelling, I'm not a chemist. Okay, so you get all these interactions happening, you get some sort of ionic type bonding as well, interactions as well, that will cause the final three-dimensional shape. So you might get, going this way with your alpha helices, a beta pleated sheet coming back on itself. Until you have your final three-dimensional protein. So what makes each protein different is this primary sequence. Primary sequence causes different amino acids to be next to each other, which therefore causes them to have different interactions which causes them to have alpha helices, beta pleated sheets in different positions, which causes different hydrogen bonding, van der Waals interactions, other interactions as well, which causes their folding, coiling, attractions and repulsions, causing their final three-dimensional shape. This is now potentially your final protein. Okay? Every protein gets to this stage. Most are done at this stage. Okay? Or could be considered done at this stage. The final stage is what, Jamie? 2. Quaternary. And this quaternary stage is where we have more than one protein interacting together. Okay? So quaternary, more than one joined together. Okay? More than one protein joined together. A classic example of this is hemoglobin, which has 4 proteins that make it up. Okay? So before it's a functioning hemoglobin molecule, it needs 4 separate proteins, coded by 4 separate genes. Okay? So you might get something like there's 1, and then there's
So once the polypeptide chain's done, it's just float around until it finds whatever it's other.
yeah. So they'll probably be directed to be made very close to one another. So there may even be additional molecules that help join them together. But basically, as soon as it's coming out of the ribosome, and if you remember that video I showed you, as soon as it's coming out of the ribosome, it's already forming those alpha helices, beta pleated sheets, it's already folding. So it's instantaneous. As soon as it's made, it makes that three-dimensional shape, and then it's ready to provide that function. Okay? So what are the functions of proteins? Enzymes. Catalysts are enzymes. Hormones. Okay, speak to Eston, speak to Mrs. McWaters. She knows all about peptide drugs. Filthy Essendon Football Club. Okay, injecting peptides into one another, having a merry old time to improve their endurance. But we have a whole range of peptide hormones. So these are protein hormones. Okay?
Antibodies.
Good, Nick. Oops. Let's go. Antibodies. So our body is making millions and millions of different types of antibodies, hoping that if something invades our body, we can recognize it, therefore we can create a response to it. So we just make all these different antibodies. What else?
Structural proteins.
Structural proteins. Okay, these can be made up or evident in ligaments, tendons, in the cytoskeleton, all different areas to provide structure. What else? Excellent. Hemoglobin. So hopefully something like that. What else? What's that stage where we have to get to be better than others? James? Create. Go above and beyond. Okay? create. Remember,
Oh,
learn, master, create.
Yeah.
So to stand out, you've got to go that a little bit above and beyond. We also have cell surface receptors. We have cell membrane pumps and channels. And we have regulatory inhibitors. That's just a few. There are lots more. So hopefully this goes back to every cell. Why does even the simplest cell have so many genes? It's because we have so many proteins that do different things. So if they're all made from the same 20 amino acids, hopefully now you can see that the only thing that makes them different is that sequence of amino acids. That's it. Same 20 amino acids. The only thing that makes them different is that sequence of amino acids will then cause them to form a different three-dimensional shape, which will then cause them to have a different function. So three-dimensional shape is so important in biology because that's what helps things interact. Okay? Makes them gel together. Questions? Easy? Okay. The types of questions that you'll get is you might be asked to explain how does a protein's structure determine its function, how does the sequence of amino acids determine its function, how does we get from this to this. They're the types of questions. Maybe you could get an extended response question. Explain the organisation of a protein, and you have to go into detail. Extended responses is where you show your create. Okay, that's where you show your extra knowledge. You can't really get a 15 out of 15 unless you're adding that little bit extra. Okay, if you're perfect. All good? Yeah, that. This is fun.